What the absolute fricking heck is this crazy G-protein stuff? Holy moly, this is some wild cellular communication. How does a cell even know what the frick is going on outside its tiny little self? This stuff is fricking nuts. So you've got this absolute legend, Alfred G. Gilman, whose dad was already a fricking pharmacology rock star. Gilman comes along and blows everyone's minds by figuring out how cells actually talk to each other. Before him, Martin Rodbell had this crazy idea about a three-part system: a receptor, an amplifier, and some mysterious transducer linking them. But no one knew what the frick that transducer actually was. Gilman, this absolute genius, uses these fricking mutant S49 mouse cells that are broken in just the right way. He mixes them with normal cell bits and bam, he restores the fricking signal. Through years of painstaking, crazy-but work, he purifies this elusive transducer and identifies it as a protein that binds to guanine nucleotides. He calls them G-proteins. He discovers this GS protein is a fricking heterotrimer with alpha, beta, and gamma subunits. The alpha subunit is the fricking star, binding GTP and kicking off the whole wild show. This crazy G-protein acts like a molecular switch. When it's inactive, it's chilling with GDP. But when a signal hits the receptor, it swaps GDP for GTP, the alpha subunit breaks off, and both the alpha and beta-gamma parts go off to regulate other fricking molecules. Then the alpha subunit slowly hydrolyzes GTP back to GDP, turning the fricking signal off. This is why this fricking stuff absolutely fricking matters. G-proteins are the targets for nearly a third of all fricking drugs out there. And when they go wrong, like with cholera toxin permanently locking GS-alpha in the ON position, you get crazy diseases. Gilman and Rodbell won the fricking Nobel Prize for this wild discovery. This nuts research laid the foundation for understanding how our bodies fricking work. Learn more about this crazy fricking science at brainrotstudy.com.